Welcome to the Depth Dive. We're plunging into the world of information today. And, the amount of data we're all creating, it's just staggering. It's growing faster and faster. We're talking petabytes, exabytes, these huge numbers. And for ages, we've relied on silicon chips, right? For storage, for processing. But what happens when silicon starts hitting its limits? Are there even limits? Well, get ready for something truly mind-bending. Because the molecule of life itself, DNA, is stepping up. It's emerging as this revolutionary new way to store massive amounts of data and even perform complex computations. Sounds like science fiction, I know.
Yeah,
But what you're about to hear, it's very real science.
it absolutely is. And in this deep dive, we're going to unpack some really exciting recent breakthroughs, how scientists are using DNA not just as, the blueprint for life, but as a programmable material for IT. We'll look at the potential, which is huge. We'll also get into the big challenges they're facing, because it's not easy, and the really clever solutions pushing the boundaries. It's a fascinating field right now.
Okay, let's get into it then. DNA. Most people, like me, think genetics, right? Heredity, that code inside us. But storing the world's data, like every text message, every cat video, every movie, in a few grams of DNA, why is that so attractive? What makes it the ultimate archive?
Well, it really boils down to two incredible things: density and longevity, just how much you can pack in and how long it lasts. DNA density as well, it's astonishing, over 17 exabytes per gram.
Think about that.
That's 17 exabytes, that's hard to even picture.
Exactly. Imagine taking a whole football stadium packed with servers, the blinking lights, the cooling systems,
Yeah.
and replacing all in that with something you can literally hold in your hand.
Wow.
And then there's the longevity. Under the right conditions, cool, dry, dark, DNA can last for thousands, maybe tens of thousands of years, way longer than any hard drive or tape. Okay, that's just incredible. Density, and it lasts forever. Practically, it really changes how you think about, like, archiving human knowledge. But, okay, there have to be big hurdles, right? How do
Yeah.
you actually get the data into the DNA? And maybe more importantly, how do you get it out again reliably?
Yeah.
Especially at huge scales. It can't be as simple as, drag and drop.
Oh, definitely not. You've hit on the key challenges. Getting data in means synthesizing DNA strands precisely, and getting it out means sequencing it. And one really big problem has been retrieval, especially with the new ways we make DNA, these high-throughput methods like chip-scale synthesis, where you make thousands of different strands at once. You end up with lots of different sequences, but some are in very low amounts, very low copy numbers.
Okay.
And when you try to amplify them using traditional methods like PCR, well, PCR isn't perfect.
Oh,
Yeah, why?
indices. Meaning what?
Meaning some DNA sequences just amplify much, much better than others. They get copied more times. So the ones that were already rare might get lost entirely or
Yeah.
become even harder to find.
So your data gets skewed. You lose bits, or maybe whole files.
Exactly. You either lose data or you have to sequence everything incredibly deeply, spend a fortune, basically just to make sure you catch those rare sequences. It makes the whole process really expensive.
Right. That sounds like a major roadblock. So this is where it gets really interesting. How are researchers tackling that amplification bias? What's the clever fix?
Well, this is where a team led by Zi Wang, Jiangshu Li, and Chunhai Fan came up with a really ingenious solution. They called it MPADs.
MPADs. Bit of a mouthful.
Yeah. It stands for Massively Parallel Homogeneous Amplification of Chip-Scale DNA for DNA information storage. The key idea was to tackle the primers used in PCR.
The primers. Those are the little starting points for copying the DNA.
Exactly. Traditionally, primers have a fixed length. But because the DNA sequences storing the data vary, these fixed-length primers bind with different efficiencies. Some bind strongly, some weakly. That's the source of the bias.
Okay.
So instead, they designed fixed-energy primers.
Fixed energy. What does that mean?
It means they designed the primers so that regardless of the specific data sequence they're binding to, the thermodynamic binding energy, how strongly they stick, is consistent across all the primers for all the sequences.
Clever. So they all bind equally well, more or less.
Pretty much, yeah. Much more uniformly, and the impact is huge. When they amplify the DNA library, all the different stored sequences get copied much more evenly. The representation is far more homogeneous,
And that solves the cost problem.
dramatically. Because everything is represented evenly, you don't need to sequence nearly as deeply to recover all the data. They estimate this homogeneity could lower sequencing costs by up to 4 orders of magnitude.
4 orders. That's like 10,000 times cheaper.
Potentially, yes. Compared to needing maybe 11,000 times deeper sequencing with the old primers just to get uniform coverage. It's a massive saving in cost and time. And they tested this. They used over 35,000 different DNA sequences, encoded multimedia files, text, images, videos. They could get them all back out. And get this, they even showed they could get decent decoding accuracy, around 80%, even at super low sequencing depths, like just 1x coverage, if they combined it with some machine learning tricks to clean up the images afterwards.
Wow. Okay, so that makes large-scale DNA storage seem much more, well, practical, almost ready for industry.
Bringing it a lot closer, definitely.
Yeah.
Removing a major economic barrier.
All right, so DNA is this amazing, super dense, long-lasting hard drive, a huge leap for storage. But you mentioned computing too. Can DNA actually think? Can it process information, act like a tiny computer?
Absolutely. It's not thinking like we do, of course, but it can perform computations. The way DNA strands pair up A with T, G with C, and how they can self-assemble into complex structures that provides a really powerful platform for what we call molecular computing.
Molecular computing. Oh, okay. How do you program that? You're not writing code like Python or C++ I assume.
No, not in the same way. It's more about designing specific DNA molecules that will interact in predictable ways to carry out logical operations or solve problems. One really cool approach was demonstrated by Zhenghuang, Mo Shi, Lilinu Yang, and their team, including Ji Chao. They built what they call an artificial molecular communication network, or DRAMCN.
Okay, DRAMCN. What does that do?
They use DNA origami, where you fold long DNA strands into precise nanoscale shapes, like tiny rectangles in this case. These rectangles act as the nodes in their network, like processing units.
Like little circuit components made of DNA?
Kind of, yeah. And then they add programmable sticky ends, short, single-stranded DNA bits onto these origami structures. These act as the edges, or connections, allowing the nodes to link up and communicate, passing signals along. They showed the system could do different kinds of communication: serial, parallel, multiplexed, mimicking how signals travel in biological systems or even in our own computer networks. It's building the infrastructure for DNA computation.
That sounds incredibly intricate, building networks node by node with DNA, and they actually used this to solve a real computational problem.
They did. They tackled a famous one, the Hamiltonian path problem.
Ah, yes, the traveling salesman's cousin, sort of, finding a path that visits every city or node exactly once.
Exactly. It's what's called an NP-complete problem, notoriously difficult for traditional computers to solve efficiently as the network size grows. Their DNA network, the DRAMCN, combined with a clever algorithm to filter out wrong paths early on, actually found the correct Hamiltonian path. And the readout was fascinating too. They didn't just get a number, they physically separated the DNA structures that represented the correct path using a technique called rate zonal centrifugation. You literally spin the tubes, and the DNA representing the solution separates out.
You spin it, and the answer just appears as a layer of DNA. That's wild.
It's a really elegant demonstration of harnessing DNA's physical properties for computation. And thinking bigger picture. This kind of work gives us insights into how communication actually works inside our cells, signal transduction. Plus, it opens doors for things like smart biomedical devices, maybe molecular robots for drug delivery or advanced imaging.
Okay, so we've gone from storage to solving abstract math problems in a test tube. Can we push it even further? Can we use these DNA computing ideas to program living cells, to engineer life itself?
That's exactly where synthetic biology is heading. And yes, recent work shows we're getting much better at engineering predictable, powerful genetic circuits inside living organisms. Two great examples, one in mammalian cells, one in plants.
Let's start with mammalian cells, our cells. What's the big difficulty in controlling genes there for things like gene therapy or making biotech products?
Well, a key challenge is getting tight control. You want your engineered gene circuit to be completely off when it's supposed to be off. Minimal leakiness, we call it. Any background activity can be harmful or inefficient. But you also want it to be really strongly on when you activate it, getting both low leakiness and high maximum expression. That's a tough trade-off. It often involves a lot of painstaking trial and error.
Right, like a light switch that's never quite off or never quite bright enough. How did Giuliano Di Carluccio, Virginia Fusco, and Diego Di Bernardo tackle this with their Caswitch system?
They did something really clever. They combined two known ways gene networks regulate themselves, something called a coherent feed-forward loop and another called mutual inhibition. Think of it like using standard electronic components but made of genes. They integrated these motifs with CRISPR technology, specifically an enzyme called Cas-Arcs, and a common on-switch system called TET-on3G. And the result, their Caswitch version 2, is pretty astounding. They managed to reduce the leakiness by more than 10 times.
Wow.
While fully recovering the maximum on state. This gave them an incredible dynamic range, a 3,000-fold difference.
Yeah.
Off and on. That's a huge leap in precision control.
3,000-fold.
Yeah,
That's like going from a dim nightlight to a searchlight instantly.
exactly. And the practical applications are immediate. They used it to make a copper biosensor 10 times more sensitive. They also show they could use it to tightly control a toxic gene, which is super important if you're using cells as factories in biomanufacturing. You don't want the product killing the factory. And for gene therapy, using viruses like AAV to deliver genes, their switch significantly cut down on unwanted expression from the virus itself, making the therapy potentially safer and more effective.
Okay, that's huge for medicine and biotech. But what about plants? You said there was progress there, too. Plants seem like a totally different challenge. They grow so slowly. Engineering them must take ages.
You're absolutely right. That slow iteration time, months, just to test one design in a whole plant, has been a massive bottleneck for plant synthetic biology.
So how did Xikong, Yin-Yang, Tiankang-Shi, and Shuizong speed things up? What was their breakthrough?
Their key innovation was developing a rapid prototyping system using Arabidopsis leaf protoplasts. These are plant cells without their tough outer wall.
Protoplasts. Okay, so testing in cells, not whole plants.
For the initial design phase, yes. This cut down the design build test cycle from months to about 10 days. A huge speed up.
10 days versus months, that's game-changing for design.
Definitely. And they also standardized everything. They created relative promoter units, RPUs, so they could reliably measure and compare the strength of different genetic parts like engineers do with volts or amps. They built a toolkit, sensors for plant hormones like auxin, logic gates like NOT gates using repressors, all standardized and characterized quickly in protoplasts.
But the big question then is, does it work in a real plant? Do the models built in these isolated cells actually predict what happens in a living, growing plant?
That's exactly what they tested. And the answer was yes. They built logic circuits, things like A imply Y, B gates or NAND gates, based on their protoplast data, and they successfully used these circuits to control where and when genes were turned on in vivo, inside living Arabidopsis roots, just by applying specific chemical signals. The predictions held up.
So they could actually program patterns of gene expression in a living plant root.
Precisely. And they took it one step further towards controlling plant traits.
Like making them healthier or more resilient.
Exactly. They engineered an OR gate in tobacco plants. If they applied chemical A or chemical B, the gate would activate an immune gene. This allowed them to trigger the plant's hypersensitive response, a defense mechanism on demand, using external chemical cues. It's programmable control over a key plant health trait.
That's amazing. Programmable immunity for plants. The potential for agriculture is enormous.
It really is. Think crops designed to resist specific diseases only when needed, or plants that can better handle drought stress based on signals.
So let's tie this all together. What does this mean for you, our listener? We've journeyed from DNA as super dense data storage
Classic millennia,
to DNA performing complex computation.
involving hard math problems.
And now to actually programming living cells, both mammalian cells for health and plants for, well, potentially feeding the world more effectively.
Yeah, if you connect the dots, the bigger picture is that biology is truly becoming an engineering discipline. We're learning to design, build, and test complex biological systems, using DNA as the code, the wire, the component. The common thread in all these studies is that ingenuity in overcoming the core challenges. Reliability in storage retrieval, scalability in computation, precision control in living systems. It's about making biology predictable and programmable.
It really does feel like we're standing on the edge of a biological revolution, storing data forever, tiny computers inside us fighting disease, crops designed for a changing planet. It's mind-boggling stuff.
And it leaves you wondering, doesn't it? What other impossible problems might DNA hold the key to solving now that we're learning its language for information and computation? What comes next?
The future of information, our health, agriculture. It might literally be written, computed, and engineered using DNA. Imagine intelligent, self-repairing data centers made of DNA, tiny biocomputers diagnosing us from the inside, crops adapting dynamically to the weather. This deep dive really shows that sometimes the most powerful, most elegant solutions are hidden in the fundamental building blocks of life itself. We just needed to learn how to use them.